Hello guys, in the previous two videos, we have completed the introduction of this book up to unit 5. So now in this video, we will start from unit number 6. Let's start with it and see what are the contents of unit number 6. So unit number 6 is all about DNA. DNA is an essential molecule for all living things. That's true. The structure and function of DNA. So 6.1, DNA structure and replication. How it replicates during the cell cycle. This whole unit is covered with respect to the cell cycle, which we have completed in unit 5. So exploration 1 is the function of DNA. So first we should know why DNA is important, what are its functions, and how it carry on the functions. Exploration number 2 will be the structure of DNA. Structure of DNA is complex but very interesting. Its ladder structure is the most believable structure and it was given by Watson and Crick. It is called Watson and Crick model of DNA. Exploration 2 is DNA replication. DNA replicates and one strand we get two strands after replication. And in the laboratory, extracting DNA. So now this is a very advanced technique, but students will do it on a simple level. Protein synthesis. 6.2 is protein synthesis. So now we have studied the function and structure and replication. Here comes the next step, transcription and translation. Now from DNA, protein is synthesized. Introduction to protein synthesis. Exploration 1. Exploration 2 is transcription of DNA and exploration is translation. These two steps are very important to make or synthesize proteins. Language arts connection making synthetic cells. How to make synthetic cells? This is an activity and evaluation will be done online by the teacher. Then at 6.3, gene expression and regulation. Exploration 1, regulating gene expression. Exploration 2, gene regulation in eukaryotes. Exploration 3, factors that influence gene expression. This is very important topic. Career in science geneticist. Okay, those who study this gene expression and regulation, protein synthesis, DNA, those who have grasp on this subject, they are called geneticists. Altogether, they are all biologists, but for this specific study of DNA and gene, they are called geneticists. Then comes the new unit review, which can be done online, and unit performance task is given to students to complete online. Now we move on to unit number 7. Unit number 7 of this book. Unit number 7 is genetics and heredity. Proteins like Cas5 can be used to edit DNA. Now DNA can be edited. You can remove or add genes into DNA. This is a very modern technique and we will study it later in this chapter. 7.1 is meiosis. Exploration 1 is chromosomes and meiosis. Before studying meiosis, you should know what are chromosomes, how they are formed, at what stage of cell cycle chromosomes are formed from the chromatin. Exploration 2 is the process of meiosis, and exploration 3 is meiosis and genetic variation. So now different organisms are different from each other. It's due to genetic variation that we will study in unit 7.1 meiosis exploration 3. This is very important. Guided research will be gene duplication and genetic variation. Evaluation will be done. And then 7.2 Mendel and heredity. So Mendel's law are very famous. How he completed his studies on pea plant. Exploration 1 will be Mendel's groundwork for genetics. Exploration 2 is traits, genes and alleles. Exploration 3 is extended Mendelian genetics. Success, career in science, genomics, study genomes. So when we study meiosis and all these genes, traits and alleles, this is called study of genome and genomics. This actually it is biology, but this specific topic is genomics. 7.3 is traits and probability. Exploration 1 is predicting generations. Exploration 2 is determining types of crosses. Exploration 3 is sex linked inheritance. Data analysis is pedigrees. We will look at the pedigrees of different animals and different families. Now 7.4 is mutations and genetic diversity. Gene mutation. There are many different types of mutations. Mutation means abnormality in the DNA. It can be useful, but most of the time it is harmful and it is not beneficent. Exploration 2 is chromosomal mutations. What happens when mutations are there in chromosomes? At what chromosomes? At what point on chromosomes mutation occurs? Exploration 3 effects of mutations, the diseases that are caused by the mutations, and then testing UV protective fabrics. Engineering concept is there, and evaluation will be done. Now 7.5, it's a long unit. Genetics and heredity is a long unit. Now let's move on to genetic engineering. Exploration 1 is isolating genes. We will study isolating genes, how genes are isolated. Cloning and engineering in exploration 2, and exploration 3 is genetically engineering salmon. Genetically engineered salmon is there, that we will study later in this chapter. Exploration 4 is further applications of genetic engineering. Language art connection, knockout mice. Evaluation is done online. Unit review is done by the teacher, and unit performance task is given to the students, and they will solve it as a summative assessment, most probably. Now, next is unit number 8. It is related to evolution. Many species develop unique features that help them survive. Now, it says that according to the environment, species change their features or develop new features. Evidence for evolution, page number 380, unit 8. 8.1 will be lines of evidence for evolution. Exploration 1 is molecular and genetic evidence. Exploration 2, developmental and anatomical evidence. Anatomical evidence, exploration 3 will be geological and fossil evidence. Very important topic, fossils. Data analysis is radiometric dating and evaluation. This is all for 8.1 from evidence for evolution. This is a very technical topic. Then theory of natural selection. This is a very controversial theory, and there is a dispute among scientists for this. So exploration 1 is hands-on lab modeling natural selection owls and field mice. There is an investigation where students will study this and answer the given questions. Exploration 2 is developing theory of natural selection, how theory of natural selection was developed over time. Then there are four principle of natural selection are there, exploration 3, and then research is natural selection today. So how you will explain natural selection today, that will be the research guide. Unit review will be done, and unit performance task will be given to the students. Students will be evaluated. So that's all for evidence for evolution, lines of evidence, theory of natural selection. It is a very, very interesting topic. And next we have unit number 9, which we will do in the next video. Thank you so much for watching. Stay in touch. Bye bye.